Hello, I am Priyanka Arora from Department of Biotechnology, Rama University. I am here to deliver a lecture on Southern blotting. So, Southern blotting, it is a detection technique and it is used to find the target DNA sequence in the known DNA sample. Okay. For example, if we are looking out for some gene rearrangements, if you have to study the gene rearrangements, or we are looking for the mutations in the DNA of the organism, we can take the sample of the organism and perform the Southern blotting technique to look if that sequence is present in the organism. Okay. So, this technique is based on the principle that first it is used for the separation of the DNA using the gel electrophoresis, the transfer of the DNA fragments onto the membrane, and then we are identifying the known sequence if it is present or not using the label probe. Okay. For this, if we look into the procedure of the Southern blotting, first we will isolate the DNA of the sample. This is the DNA of the sample which can be isolated from the eukaryotic or the prokaryotic organism. Okay. The known sample, in this sample we are looking for the known sequence if that sequence is present in the sample or not. Okay. After that, we will digest this sample using the restriction enzymes. Restriction enzymes are the endonucleases which digest the DNA in the known site, which is known as restriction site. So, this DNA will be digested into a smaller fragments like this, a double stranded smaller fragments which could have our gene of interest, that is the known sequence which we are looking onto it. Okay. After that, we will perform the agarose gel electrophoresis. Agarose gel electrophoresis will perform using this using these fragments. This is the gel, agarose gel. Since we are analyzing the DNA in this sample, the DNA is separated using the agarose gel electrophoresis. Okay. We have loaded on the sample into this gel and we will get the bands which is separated on the basis of its molecular weight. Okay. Since we have to perform the blotting experiment in this technique, the word blotting comes from the word blot, which means the transfer of these DNA fragments onto a membrane. Here we are using a nitrocellulose membrane or a nylon membrane. The nitrocellulose or the nylon membrane are the membranes which have the binding affinity with the protein or the nucleic acids. Okay. So, here to perform the blotting experiment, we need a single stranded DNA. So, before moving further, we have to convert this double stranded DNA into the single stranded DNA. We can do that using giving it a mild alkali treatment. So, for this experiment, we will incubate the gel in the presence of mild alkali treatment, alkali such as NaOH, and after some time we will see that on the gel all the double stranded DNA will be converted into single stranded DNA. How this happens is in the presence of too much of NaOH, the hydrogen bonds between the DNA fragments, they get separated and we get a single stranded DNA which is present on gel. That is the agarose gel. Okay. After that, we will move on to the blotting technique. For the blotting technique, we will take a tray and place a transfer buffer onto the tray. We have a transfer buffer on a tray. After that, we will take a we'll take some either a sponge or a solid support, a glass onto the tray, and we will have a gel over our solid support. Above the gel, we will place the membrane of the same size. So, this is gel and then we have a membrane. The membrane could be nitrocellulose membrane or the nylon membrane. Above that, we will place several Whatman paper or the paper towels and we will put a weight. We will put a weight so that the gel gets in contact with the membrane. Okay. On passing the electric current on this tray, we will see that the transfer buffer will move up using the capillary action. Okay. While moving up, DNA present on the gel also moves along with the transfer buffer, and it will move onto the membrane. Since the membrane has a binding affinity for the nucleic acid, the DNA will bind onto the membrane. Okay. When we will see the Whatman paper or the paper towels get wet, we will understand that the transfer process has been completed. Okay. So, after that we will take out the membrane, we will find a membrane which is having exactly same DNA patterns, the DNA band patterns as it is present on the gel. So, why are we actually doing this blotting technique? We are doing this blotting technique because we cannot use the gel for further analysis of the DNA. As agarose gel is a very fragile, to take it onto another membrane, we have used the blotting technique and placed the similar bands onto the membrane. Okay. So, the bands which is present on the membrane will not be visible for now. So, we will give it we will fix the DNA onto the membrane by using the heat treatment, by giving the heat treatment or UV light. Okay. We will bake the DNA into the membrane. After that, we will place this membrane onto a plate which is having a probe and incubate it for some time. This is the membrane. Okay, onto a probe, and we will let it incubate for some time. So, what is probe? Probe is a short oligonucleotide sequence which is having a detector attached to it. Okay. The detector, it could be a radio labeled atom or it could be a fluorescent molecule which is attached at the end of oligonucleotide sequence. So, this oligonucleotide sequence, the probe which we have designed, is complementary to the DNA sequences which we are finding in our sequence, in our organism. If we are looking for the organism, the sequence which is rearranged or the mutated DNA sequence, we will design a complementary probe to it, we will attach a detector to it, and if that DNA is present, if the sequence complementary to the probe is present in our sequence, which we have taken from the organism, it will hybridize with it because it is a single stranded DNA right now. Okay. So, if the gene of interest is present, it will hybridize. Thus, this experiment, this southern blotting is a hybridization based experiment or a hybridization based technique. Therefore, the probe, the label probe which is used for hybridization onto the gene of interest. Okay. So, after that, if the hybridization happens, then we will look up for the signal using autoradiography or UV light. If we have used radio labeled probe, we will look up for the signal in the autoradiography or the X-rays, or if we have added the fluorescent probe, then we will look up in the UV light. And if we get the signal, that means the gene of interest was present in our sample or the sequence which we have taken out from our organism. Okay. So, this technique is known as southern hybridization technique or the southern blotting technique, because in this technique we are only analyzing the DNA. In this way, if we are analyzing the RNA, then it is known as northern blotting technique. If we are analyzing the protein, then it is known as western blotting technique. Okay. The discoverer of this technique was Edwin Southern, and on the name of the discoverer, this technique has been named. It has many other applications, such as it can be used in the forensic sciences, it can be used for the detection of cancerous cells or cancerous sequence of the DNA, of the genetic sequences. It can also be used to study the bacterial or the viral infections. Thank you.